There was no very clear answer. This is largely because animal models, they do not have the severe cardiac phenotypes that we see in patients. And so now I'll move on to some of my research. And so these mouse models that lack dystrophin, they actually have heart problems at a very old age, over 16 months of age, which is comparable to a geriatric person, whereas in patients, heart problems can be observed even at the age of 10, where there's fibrosis by MRI, and then patients are dying in their 20s and 30s. And then the mouse models also have a mild muscle weakness. They do not have curvature of the spine, and then they live to be a normal lab mouse, living to the age of 2 years. And some of the limitations in learning about the cardiac efficacy is that in the clinical trials, if the patient has any indication of heart problems, they are excluded from the study. And in addition, it can take years for heart problems to develop, and progression is much slower in the heart in terms of functional decline. And so the clinical trials that last 2 to 5 years may not capture this efficacy, especially in a small population. And so I'm using human-induced pluripotent stem cells, where you can convert cells from the blood that are derived from the patient and have the same genetic makeup as the patient, convert them into stem cells, and then now we have all types of recipes to make different cell types. And the cell type I'm interested in is the heart. And these heart cells are really fun to make because they beat spontaneously in a dish as sheets of cells on day 10. And then later, if you keep them in a dish too long like this, then they completely lift off the plate. And so we dissociate them from each other and then replate them. And we're able to test in a dish whether these gene therapy strategies are effective. And today I'll be presenting data from stem cells that are derived from 3 different individuals, and then basically testing. It's a clinical trial in a dish. And so at day 10 of differentiation from stem cells, we introduce either an empty vector, so equivalent of a placebo, or the gene therapy, and it is one of the 3 miniaturized microdystrophins, and then test in these Duchenne cells where the dystrophin mutation exists, and then alongside it are the healthy control cells where the dystrophin mutations are corrected. And then some of the striking phenotypes that we see in a dish is that the cells have this much smaller morphology. So they are a lot smaller, their nuclear size is smaller, and then the staining here in green is one of the sarcomere proteins. It helps the cell contract, and the density of those proteins is lower in Duchenne. And then we can also electrically stimulate the cells. And when we electrically stimulate the cells so that they beat every 1 second, typically what you should see are these really beautiful calcium traces, where there's a nice spike of calcium every 1 second. With Duchenne, you start to see deviations from these 1 second intervals. You also see these double spike peaks that are indicative of arrhythmias. And so we see a higher frequency of these strange or arrhythmic traces. And then also we can measure their force of contraction, and it's lower in Duchenne. And then when we track the survival of the cells over time, then we also see that the Duchenne cells have a shorter lifespan. And so in the comparisons, we are comparing an empty vector control versus one of the microdystrophins, and then comparing whether we can reverse some of the phenotypes and bring them back to healthy control levels. And some of the transgenes, so the genes that are delivered, the unique properties is that this one recruits nitric oxide synthase, this one has an extra spectrin repeat that gets anchored to the plasma membrane, and this one has an extra spectrin repeat that binds the microtubules. So all of them have this N-terminal actin binding domain. They also have this cysteine-rich domain that gets anchored to the plasma membrane, but there are some distinct and unique properties of these genes, and that's where I'm trying to make the comparisons. And just asking whether these different disease characteristics can now be reversed or made a little bit better. And so far, we find that despite expressing the microdystrophins, the cell size remains smaller, and then the cardiac troponin T density is quite variable depending on the microdystrophin, but in one of the cases, it seems to improve it somewhat. And then for the calcium measurements, we capture these different traces. Again, we can capture these early after depolarization events that are indicative of arrhythmias. And largely with the healthy cells, we see a higher incidence of these healthy traces. So when you're looking at these graphs, gray is healthy, and that's what we want to see. With the Duchenne cells, you start to see these more aberrant traces, and then with the delivery of the microdystrophins, you see a bump in that gray bar. And depending on the Duchenne line, you see various outcomes, and there doesn't seem to be any consistent trends among the different comparisons. And then, but despite having some of these arrhythmic problems, they do help with survival in a dish. And so this is a cell viability assay where we compared day 40 and day 30 cells that were viable in a dish, and that the delivery of the microdystrophins helped with the survival of the cells. And this project is still ongoing, and we are continuing to measure whether the microdystrophins can help with the mechanical contraction of the cells. So this is a traction force microscopy where we cast these hydrogels with green fluorescent microspheres embedded in them. And by tracking the displacement of the microspheres, we can, and then based on the stiffness of the gel, we can calculate parameters of contraction like force, velocity of contraction, velocity of relaxation, and synchronicity. So we hope to learn more about how the microdystrophins are helping. And we also are interested in delivering larger forms of dystrophin. And so there are new strategies to basically deliver two components of AAV and then have these two independently expressed protein units spontaneously ligate together in the cell. And so we're exploring this strategy. Elaine, sitting back there, is working on that. And then we're also exploring the delivery of full-length dystrophin by mRNA, but mRNA delivery would be a transient approach, it would not, it would require multiple dosing. So we're actually going to test what type of dosing is required to reverse some of the heart phenotypes. And then we're also using patient data, so trying to correlate the mutational status of a patient with their cardiac function. So this is echocardiograms, ultrasound of the heart, and then low ejection fractions indicate that the heart is functioning poorly. And so if we can make correlations between the mutation and these low ejection fractions, we can maybe infer more information about which domains of dystrophin might be important. And so in summary, microdystrophin gene therapy has shown promise in the skeletal muscle, but little is known about efficacy in the heart.